Hi, everybody. We'll just give it maybe 1 minute to allow the last few stragglers to come in, and then we'll get started. Alrighty. Well, hello, everyone, and thank you for joining this session on integrated surveillance of malaria and LF. My name is Ashley Souza, and I will be moderating this session. Today, we'll listen to 4 brief presentations: the case for integrated surveillance in the Philippines by Betsy Miranda, results of serosurveillance of malaria and NTDs in the Philippines by Malu Mascalino, the role of entomology in integrated surveillance by Jason D'Souza, and the application of freedom from infection to support program decision-making by Jillian Stressman. During the presentations, please insert any questions you may have in the chat. If we can, we'll take a moment to answer a question or two after every presentation. The presentations will be followed by a 40-minute breakout activity. There are 3 rooms you can choose from, each chaired by one of today's presenters. Room 1 will focus on the program perspectives. Room 2 is on serosurveillance and xenomonitoring. Room 3 will be on the freedom from infection model. And then at the end of the breakout period, we'll come back together as a group, and the breakout rapporteurs will give a 3-minute presentation each, summarizing the discussion that occurred in each room. So as a heads up before we get started, some of the video presentations we're about to hear are a little bit quiet. So I would suggest that at this point, everybody maximize the volume on your computer so that you can hear and you don't miss anything. So without further ado, I think Dr. Betsy Miranda will get us started.
Thank you very much, and good evening, good morning, everyone. May I request for the recorded video of this presentation? Good morning, good afternoon, good evening to all the participants. I am Betsy Miranda, and I will present the case for integrating the surveillance of malaria and lymphatic filariasis in the Philippines. Great achievements have been made by the national programs for malaria and lymphatic filariasis, but elimination of these diseases remain elusive in the Philippines. The country was first in the Asia-Pacific region in 2001 to implement MDA for LF. It was one of the first 10 countries in the region, which in 2009 committed to malaria elimination by 2020. The country has steadily moved towards the goal of all 44 LF endemic provinces in the post-LF MDA status by 2020. However, for the past 5 years, post-MDA provinces have experienced outbreaks and others are struggling to reduce disease prevalence. Although the number of malaria cases nationwide has steadily decreased and is now reported in only 3 of 81 provinces, the goal of elimination has been pushed back to 2030. The reduction in cases in the provinces of Palawan, where 98% of the cases are reported, has stagnated since 2016 and is observed to be slowly increasing, and outbreaks have been reported in the remaining 2 provinces over the past years. The national programs of malaria and LF need more sensitive evidence to guide their decision-making, as these diseases near elimination status. Why integrate surveillance for malaria and LF? One reason is that in the Philippines, these two diseases share a common vector, Anopheles flavirostris, and are co-endemic in provinces that still report these diseases. However, there are provinces with these two diseases but QLEX as the vector for LF. The table on your right hand shows the provinces with these two diseases and where LF, schistosomiasis, malaria, and other diseases are targeted for 2030 elimination. For another reason, in January 2021, the Department of Health announced guidelines on establishing integrated elimination hubs for malaria and lymphatic filariasis, with surveillance for cases and vectors as the key activities. For malaria, a province will not be declared malaria-free unless it has an operational malaria elimination hub, among the other requirements. As of today, there is no province with any integrated elimination hub for these two diseases. Last year, the Philippines was fortunate to obtain a grant to investigate how malaria and LF surveillance can be integrated to improve decision-making to accelerate the elimination of these diseases. We propose to develop an integrated and enhanced surveillance of malaria and LF to generate the following information. 1. The presence of exposure to or infection with malaria and LF. 2. The absence of transmission or recrudescence of malaria and or LF. 3. To identify the foci of infection or transmission and the likelihood of detection by surveillance. Because of the heterogeneity of transmission of malaria and LF, we realize the importance of understanding the information that will be derived from the tools and approaches we use and is illustrated in this slide. In a general sense, the interpretation of information is dependent upon the phase of the transmission of the disease. These tools and platforms and information derived from them will be discussed by the next three speakers of this session. I wish to point out that although our project does not include xenomonitoring, there is an area where we hope to explore. A reason why we would like to study this approach in the Philippines is that in response to the COVID-19 pandemic, the Department of Health is undergoing re-engineering to improve or repair its health systems for universal health care and to better respond to pandemics and emerging and re-emerging infections. Disease surveillance at all levels of the health system is a backbone for disease surveillance, prevention, control, and elimination. For objective 1, we want to identify the gaps in the surveillance of malaria and LF for disease elimination and generate new data and interpretation of old and new data. Our questions are, has the transmission been interrupted? Is the area disease-free? Is there resurgence of diseases? Our second objective is for program management, including processing of new information from enhanced surveillance. The data and information management from the Centers for Health Development and the Central Office of the Department of Health and the local government units, and the joint administration of surveys for malaria and LF. This work was the result of a long partnership among our research collaborators and partners, including the Global Initiative for Disease Elimination, or GLIDE, with the Falcon Awards, the primary recipient, which is the Research Institute for Tropical Medicine, local and global collaborators like FETPAFI, LSHTM, the University of Southern California, the University of Glasgow, and of course, our national partners from the Department of Health and the Regional Health Department. Thank you very much.
Good day, everyone. I'm Maluma Kalinaw from the Research Institute for Tropical Medicine, Philippines, and I'll be talking about the potential application of multiplex serology for integrated disease surveillance. As we all know, countries are committed to achieve the goal of elimination for malaria and NTDs, including lymphatic filariasis by 2030, and that includes the Philippines. So for this, we are trying out different strategies to eliminate these diseases. And today, I'm going to focus on one of these novel approaches, which is multiplex serosurveillance. So serology is the measurement of the antibody responses to pathogen-specific antigens in a sample, usually blood sera. And through this, we get an idea of the exposure of an individual to a particular pathogen. In malaria, it's considered to be a proxy for entomological inoculation rates, which are the direct measurements of malaria transmission. But then disadvantage of serology is that the utility of serological markers need to be validated in different countries and different endemic settings. Multiplexing assays allows for the simultaneous analysis of different analyses in a single run, which means you don't need to run multiple ELISAs. It also means you can get more data from our samples, but as with any assay, it needs to be optimized and if possible, standardized. We wanted to test the potential of serosurveillance, and so our ITM had a collaborative project with LSHTM called Ensure, where we explore different strategies for enhanced surveillance. For this study, we had 3 sites, so Bataan, which reported no local malaria since the 90s, Occidental Mindoro with limited exposure, and Palawan with ongoing transmission. We conducted health facility-based surveys where we recorded participant data, including geospatial data in tablets, and collected dried blood spot samples from health facility attendees and their companions for molecular and serological surveillance. For serology, we used the Luminex platform for the multiplex bead-based assay, which allowed up to 100 analytes per sample per run. Our malaria serology panel consisted of 8 PF and 6 PV blood stage antigens. And when we analyzed their correlation with PF and PV infection, we found a higher association of PF infection with PF markers and PV with PV markers, showing the specificity of these markers, particularly the ones highlighted, which are the recent exposure markers. Of course, our next concern is how to interpret our data. And first, we graph our raw data, reported as net MFI values. And we can already see from this single antigen, PFAMA1, that the antibody response levels are consistent with the known status of malaria transmission in our sites. So Bataan always had the lowest, Occidental Mindoro next, and Palawan always had the highest antibody levels for all of the age groups. We also used different analytical approaches to classify recent or historical exposure. For the single antigens, we determined seropositivity cutoffs using different models. And we also did multiplex analysis of the data through machine learning models. For results, for the PF recent exposure, we found that as expected, using a single marker such as eChamp5, which is a recent exposure marker, can show some background seropositivity. But this was improved with machine learning predictions, which showed 0 to almost 0% recent exposure prediction for Occidental Mindoro and Bataan, which agrees with what we know about their status. Our serological findings can also be linked with actual infection data and also geospatial data to make maps. And as an example, here is a small area in Palawan showing the location of malaria cases in specific months during our survey. And as you can see, case locations differed per time point. but the serological data is able to provide a snapshot of these recent infections in a single collection, and comparing the seropositivity of the historical markers in orange gives us more information about the progress of transmission in the area. Another example of an application is the mapping of our 3 study sites with different transmission intensity. And based on our survey samples, you can see that there are mostly reds in Palawan and Occidental Mindoro. But when we map just the survey participants age 20 and below, we can clearly see the reduction of seropositives in Occidental Mindoro, and there's almost none in Bataan, suggesting the absence of PF transmission in this site. So this can potentially inform elimination programs on the progress of their efforts. So next, to make the most out of multiplex serology, we are also analyzing the ENSURE samples antibody responses to NTD antigens. So this includes STH, LF, dengue, etc. And shown here are maps of the antibody levels of these LF antigens. And as you can see, they had varied levels. And Palawan has been declared LF-free in 2015, so these likely reflect historical exposure. And we can probably compare this with historical data. We also determined the seropositivity cutoffs for those LF antigens. And in this map, I merged the data points to see if they collocate, and it seems that they do. So the next question is whether they relate with infection history and vector surveillance data. With our data, we can also see the distribution of malaria seropositives in relation to other diseases like LF and dengue. And we are excited to see if there are trends that we find out. So our key takeaways then are that the simultaneously collected data from multiplex surveillance has the potential to inform these disease elimination programs in co-endemic areas, for example, malaria and LF, which we are exploring in our ENSURE 4.2 project. This assay can use dried blood spot samples, which makes it easier for field collection. Also, serological markers can inform of the absence of transmission of diseases of interest in elimination areas. And lastly, the multiplex data can be analyzed in various ways depending on the information you need, and can be useful for an integrated surveillance approach. So with that, thank you for your attention.
Hello. Good morning, good afternoon, and good evening, depending on where you are. My presentation is on the opportunities for integrating malaria and LF surveillance activities, but from an entomological point of view. Both malaria and LF are vector-borne diseases targeted for elimination, but with different characteristics and time points. However, they both have similarities in that in many settings, they are both transmitted by Anopheles mosquitoes. The role of entomology in elimination is therefore very important for vector-borne disease surveillance. For malaria, entomology plays a role in insecticide resistance monitoring, transmission, and infection surveillance. For LF programs, entomology is also important to inform if programs have reached the endpoint of NTD elimination and provide evidence that transmission is interrupted. Entomology is also instrumental in ascertaining hotspots in post-intervention or surveillance areas. Fortunately, we have many tools and practices to learn from and to optimize in order to efficiently meet the needs and challenges facing the NTD entomology community. Here are some examples of manuscripts that I and some collaborators worked on that reflects examples of disease integration. These papers, although published for malaria and LF separately, have used the same mosquitoes collected in the same exercise. In Guinea, for instance, water bodies were prospected to identify mosquito breeding sites for 6 days, each in the dry season and rainy season, using the dipping method. Adults were subsequently collected from 15 communities in 5 districts in Conakry, using exit traps and indoor spraying catches over a 1-year period. Molecular approaches were employed for identification of Anopheles species, including Anopheles coluzi and gambi. Individual mosquitoes were tested for Plasmodium falciparum and Plasmodium vivax using the vectors malaria panel assay and an enzyme-linked immunosorbent assay. A proportion of mosquitoes were analyzed for W. bancrofti using dissection, loop-mediated isothermal amplification assay, LAMP, and conventional PCR methods. The results indicate that while no Anopheles were positive by the sporozoite ELISA or the VEC test, some were positive for W. bancrofti using the LAMP method. Culex mosquitoes collected during the process were also positive for W. bancrofti using both LAMP and PCR. In Côte d'Ivoire, there were similar vector collection activities as in Guinea. The collected mosquitoes were identified morphologically at species level, and some members of the Gambi complex were separated using the sign PCR method. Anopheles gambi, Anopheles funestus, and other Anopheles species were analyzed for malaria Plasmodium parasite detection using the cytochrome oxidase 1 gene. Anopheles gambi and Culex females were also analyzed for W. bancrofti infection using the PCR method. Again, in Côte d'Ivoire, we see that some parts of the Anopheles collected were positive for both W. bancrofti and P. falciparum using molecular methods. Non-vector Culex species collected during the activity were also positive for W. bancrofti. So just very quickly to conclude, we see that malaria programs collect many mosquitoes that could be used to assess LF infection in sentinel sites. LF programs may not have the technical personnel for xenomonitoring, but could benefit from malaria work in many ways. These studies indicate that the same mosquitoes, or part of the same mosquitoes collected for malaria surveillance activities, can be used for LF surveillance. Other non-vector mosquito species collected by programs can also serve LF surveillance activities. For both diseases, the same vectors and collection tools are used. Sample processing steps such as DNA extraction can be adapted based on the intended test. For ELISAs, the supernatant can be collected when the mosquitoes are crushed, and the pellets can be used for DNA extraction. There is a need to engage malaria programs at the national level to collaborate and share best practices regarding entomology, training of entomologists, and community members for basic entomology for mosquito collection. Also, LF programs can participate in mosquito collection surveys as part of malaria control program activities in order to acquire experience and use some of the mosquitoes, especially the Culex, that are usually not used by malaria programs. So briefly, these few studies show that integration between LF and malaria programs can be done. However, it requires discussions with different programs at different levels. Thank you very much for listening.
Hello, I am Jillian Stressman, and today I'm going to talk about our project on freedom from infection, a tool that we're trying to determine whether it can be used to support malaria decision-making for elimination, but something that could be equally applied to lymphatic filariasis or other diseases of public health importance. Elimination is difficult to prove because it is impractical to really confirm that there are no infections in a population unless we sample everybody in the population with the perfect diagnostic test. Otherwise, we can't be sure that the person we missed or infection that we missed doesn't contribute to transmission, and therefore elimination is not achieved. The current guidelines for elimination are disease specific. For the case of malaria, elimination means that there has been no reported cases over the previous 3 years and that there is a strong surveillance system in place to detect any reintroductions or imported infections or emergence that may happen. So the malaria elimination system really relies on the passive surveillance data available to inform decision-making. For the case of lymphatic filariasis, elimination means that infections have been sustained below the transmission assessment survey thresholds for at least 4 years after stopping mass drug administration. Therefore, for LF elimination, decision-making relies on active surveillance as part of the task surveys. So having better evidence-based approaches to support decision-making in the context of elimination would be helpful. Some of the key points are thinking about the effectiveness of the surveillance system at detecting disease transmission, but also thinking about whether the populations that are not sampled or not included are relevant and could invalidate claims of elimination. So the Freedom from Infection project was developed based on methods used in veterinary epidemiology. In that context, veterinary epidemiologists need to confirm that there's no diseases that pass through borders as part of animal trade. And so they've really developed these tools that can be used to support a quantitative decision-making process and involve estimating the sensitivity of the surveillance system for detecting the disease of interest or infection of interest. It can be used to track progress towards elimination. It also has the potential to combine different sources of information. And ultimately, all of this information comes together to estimate absence of transmission with known levels of certainty. So in the context of the Freedom from Infection framework, elimination means that if there were infections at levels at or above a predefined threshold, your surveillance system is sufficient to detect them. So if you don't detect any infections, you can be confident that there are fewer than those infections available in the population. So with this, we've tested the Freedom from Infection framework for the context of malaria. And the aim is really to see if the FFI tools can be suitably adapted for this context. We've been working with several different countries, each of which has their own unique surveillance system, so we can determine how to make the tool flexible enough to handle the differences that we are in both surveillance systems, but disease transmission patterns between countries, and also different kind of unique components of the epidemiology are reflected. The study involved collecting new data, but also model development component. In terms of the data collection, this involved retrospective collection of the routine passive surveillance data on malaria. This included information like the size of the catchment population, the number of people attending the facility per month, the number of people tested for malaria, and so forth per month. We also conducted health system interviews to get some qualitative ideas for what factors influence the performance of the health system, but also, active case detection, where we did some sampling in populations to learn how to combine those data sets. In terms of the model development, we adapted the framework developed through the veterinary epidemiologists to create a more data-driven approach so we can estimate the sensitivity of the surveillance system, which is the first step in converting that estimate to the freedom from infection. We've also worked to combine the different data sources. So for example, the active case detection or the community surveys that were conducted, integrating information from community health workers that are conducting community case management, and also thinking about how to combine information based on different diagnostic tools. So for example, infection markers and exposure markers. So this is just some quick examples of the results that we get. And so to estimate the sensitivity of the surveillance system, we need to reconstruct this scenario tree or the cascade in which an infected person goes through the health system to get detected. And then within the model framework, we can identify the probability that somebody passes through each of these steps and the factors that are associated with that. So for an example here, the probability that somebody's tested can be calculated. And then also we can see that whether they have functioning microscopy, trained staff, and no shortages, or also modify or impact the associations in the probability that somebody is tested. Just looking at some examples of malaria this is in the Philippines, where we can estimate the sensitivity of the surveillance system, which is based on this curve here. And when this blue curve here matches the red line here, we can say that a system has a strong surveillance sensitivity. This is then translated into the estimated probability of freedom. And so in this case here, we can see that there's a lot of potential cases going on in the community that just aren't detected by the health system, because this health facility in particular didn't detect any cases over this time series. And then we can see this translates into a very low probability of freedom. When the sensitivity gets better, this jumps up, but then it goes straight down because the gains are not sustained. We can also combine different sources of information. So in theory, this is a result of a simulation where if you have your information from your passive surveillance system, the probability of freedom kind of increases slowly over time. When the active survey happens, it jumps, and then it continues and jumps every time a survey happens. In terms of the data, we can look at the combination of the routine passive surveillance as well as the community health workers and the estimates of sensitivity we get from the health facility alone or the combined information. We can see this on the bottom plot where the darker green line shows the probability of freedom when you combine the two sources, and the light green line shows if it was just the health facility data alone. So we can actually calculate and estimate the added value. We can also map the probability of freedom over space and over time. So this bottom plot shows how the probability of freedom evolves over time per health facility catchment areas. And we can see some clear spatial patterns with higher probability of freedom in the northern part and slightly lower probability of freedom in the more southern part of the area. We can also look at the heterogeneity on a more local scale. So this is work in the Dominican Republic, where we can see that across the country and even within the capital city, there's quite a heterogeneous distribution of the likelihood of achieving elimination. And so in conclusion, there's a lot of potential for this freedom from infection framework, both around estimating the sensitivity of the surveillance system, particularly when relying on passive surveillance and routine data collection. We can also think about how to combine different surveillance streams and estimate the added value of each. This then translates to, we can actually estimate the probability of freedom with known certainty, and there could be a lot of potential as an integrated surveillance platform for lymphatic filariasis or other diseases of public health importance.
All right, well, thanks to our speakers for those great presentations. Before we move into the breakout sessions, we wanted to pause here to see if there are any questions for our speakers. So if you'd like to ask a question, please raise your hand or put it in the chat, and we will handle those. We've got about 9 minutes for questions.
I have one for Malau. This is Jordan. The question I have is on the LF serology for BM33 and WB123 and BM14. Are you able to correlate those positive findings with what you're actually seeing through microfilariae night bleeds? Because the serology could be positive from exposure, years ago, and I'm just curious if you're able to correlate, your serologic multiplex work against what might be recent transmission. Thanks.
So thank you for the question. So actually, right now this is still an ongoing study. So we're still gathering the data for LF in that area, and yeah, so I don't have an answer to that, but hopefully we will be able to actually relate it to the the LF transmission there. So what we know is that those antibodies are really more for long-term exposure or more for cumulative exposure. So we can see that one of them has higher antibody levels. So yeah, hopefully we can relate to it. Thanks, Jordan.
We have any other questions?
I guess a similarly related question for Z on his presentation on using xenomonitoring and getting some positive hits for LF with the same mosquitoes that are also, associated with transmission of malaria. Do you have a sense yet of how important it will be to identify the threshold for what it means to detect a positive signal in a mosquito? Like, if we have a positive signal, do we assume there's ongoing transmission and we need to continue to do, pre-tasks or task-like surveys until that signal goes away? Or is that a wide open field that we have to work our way and sort our way through? Thanks.
Thanks, Jordan. I think that's one of the questions that's been asked severally in different meetings. What is that threshold at which we can confidently say there is transmission ongoing? But detecting a positive mosquito in a community, especially post-MDA, clearly tells you there is a person in that community with infection. And that can be the starting point to go into a community and do more detailed surveillance and further activities. So about the threshold, I think we'd have to look at it again. There have been a couple of review papers by Price and Raymer, if I remember correctly, and they tried to look at different studies and infections reported in humans versus vector populations, and came up with some estimates, but those need to be confirmed.
Great, thanks, Zee.
Last call for questions. Alrighty, well, thanks to all of our speakers for the great presentations again, and thanks for the questions, Jordan. I think you've given us a lot to think about as we get ready to head into the breakout rooms. So as a reminder, there are 3 breakout rooms. Sorry, I'm just trying to get to slide share mode. And you are going to be asked to choose your breakout rooms. So the first room is going to be focused on operational research topics for integrated surveillance from the LF and malaria program perspectives. The room 2 is going to be OR topics related to serology and xenomonitoring for integrated surveillance. And then room 3 is going to be OR topics related to the use of freedom from infection tools for integrated surveillance. And so hopefully your Zoom looks something like my Zoom. So if you go to the bottom, you should see the breakout room box, and hopefully you'll have the ability to choose your room. You will have 40 minutes in the breakout rooms, and then we'll be brought back together for a brief readout on each of the discussions. So please go ahead now and select which breakout room you want to participate in. And then, like I said, you can do this by selecting the breakout room at the bottom of the screen. Note that each room does have a cap, so if we are above the limit in any particular room, we might have to shuffle some people manually. So I think the breakouts will open in about 2 minutes. So please make your selections now, and I look forward to coming together again in about 40 minutes.
Great, like integrating bednet distribution with MDA. And then there were also comments about at what level of integration should it be. And it was mentioned that it should be from top to bottom, and what mechanism. And at country level, it is very important to discuss among program managers how to integrate, and also at the national level to integrate surveillance for malaria and LF, especially including co-funding and co-management, and then also identify overlaps between these two diseases and also overlaps in identification and monitoring, including monitoring of progress. And then there was also a mention that especially for seasonal malaria, how about exploring integration of chemopreventive activities, which are quite similar with LF MDA. So I guess those are for question number for question number 1. And then for question number 2, what resources are available for integration? There was a mention of funding, especially for assessments, and then training of technicians for multiplex testing. It was mentioned that political commitment, of course, is very important. And then there is also a mention of the need for clubbing or forming of teams, presumably of the different types of workers for LF and malaria. And then also cost sharing mechanisms for LF and malaria surveys and monitoring in a country-led elimination program. And then another very important mention was about data systems to ensure the quality of data. And then for question number 3, which are the operational research topics, cost effectiveness of integrating surveillance of these two diseases, and then what mechanisms for collaboration or how to integrate. Example is malaria SMC plus MDA. And then how about looking into community acceptance of integrating these two diseases. And number 4, how can disease-specific responses be implemented at different levels of the healthcare system? And number 5 are what are the resources available for integration? And number 6, how about integrating LF diagnosis with malaria diagnosis in a post-elimination or surveillance context? And the last one was identifying target groups and sample sizes for integrated surveillance. I guess I think that's what we have in our group. And I hope I captured almost everything.
Great, thanks, Jenny. It sounds like you covered a lot in your group. So as Lee just reminded us, we have 5 more minutes until this breakout session ends. So I will pass it over quickly to Millicent for a readout on group 2.
So for my group, we were supposed to look at integrating surveillance for malaria and lymphatic filariasis. And the first question we had to deal with was key research questions that should be prioritized. And so for this, we're looking at identifying sentinel sites for both diseases, so in the discussions, we talked about the fact that we need to identify sites that will have both diseases present. And then if we are analyzing samples, then we are sure that we are going to get positivity in those samples. And then for seropositivity, we know that for malaria, there are several markers for detecting current infection. However, for LF, these are not available. And so what does seropositivity for LF antigens mean? Are they current or past infections? So I think we can learn from malaria and also develop markers that can help us detect current infections. And also, looking at both diseases, we know there are different dynamics. Whereas malaria could be as a result of a few bites of mosquitoes. LF, you need several bites. And so if we are integrating this, we need to know how best to draw conclusions from the results we get. Doesn't mean that if you test a few mosquitoes and you get positive for such mosquitoes, they translate into transmission of LF. I think those are some questions that we talked about. And for current tools that are available, we know there are serological tools for both malaria and LF. So we're thinking of multiplexing. So there's Luminex assays where you can put a lot of targets, and that is being developed for LF, malaria, and some other viral diseases. So if we can incorporate multiplexing of some of these serological assays, that would help. And also, there was also the mention of using cell-free antigens. So there are instances where you could find some antigens circulating in the blood of humans. So if these parasites are blood parasites, there could be some antigens that can be detected using both samples. So whereas people, malaria control programs may use the samples for detecting malaria, these same samples can also be used for screening for LF. And then opportunities. So what are opportunities for integrating malaria and LF surveillance? So currently, malaria programs collect blood from people, but then there was this question of whether kits are used. If kits are used for such surveys and you detect positivity in these kits, does it translate into ongoing transmission for LF? And I would say yes, because for LF, in order to pass to the elimination state, then children would have to have cleared infection. So that's why when you detect infection in children, then you need to go back and survey the community because it signifies ongoing infection. And so for the opportunities, we basically talked about the fact that we can use samples collected for malaria surveillance, for LF surveillance, so in terms of blood and also vector samples. Thank you.
Thank you, Millicent. And then Nico, group 3, last word, and you have about 1 minute. Hopefully we don't get cut off.
Okay, great, thank you. Well, given this, for the sake of time, I'm just going to cut straight into the knowledge gaps and recommendations for next steps. We'll reduce it essentially down to 3 big points. The first being how to move from only routine data collection. For example, malaria, this is widely available, but in the case of LF, it might be more difficult to have sufficient data for the modeling. And for the LF, for the TAS surveys, it's typically in a low density coverage population. Since the modeling can be done as an iterative process for the freedom from infection framework, we can look at settings where we can have this data that's been collected multiple times to better understand applicability. Another important note that was mentioned was that when determining the geographic scope of the estimates, this is going to be different between diseases and context. So that's an important consideration when thinking about the application of the freedom from infection framework. And I think I'm at time. Is it going to cut me off immediately or?
Keep going. Last point. Go ahead.
Okay, perfect. The last point is kind of just an overarching thought in that we need to be realistic, especially when we're talking about working with post-data collection frameworks, that we should work with what's available and not let, I think that the phrase used was perfection become the enemy of progress.
Awesome. Well, thank you for the really succinct descriptions of everything that was discussed. It sounds like all of the groups had really productive conversations, and I'm looking forward to seeing the outcomes written up. At this point, the session is over. Quarantine Day 1 is over, but we are looking forward to hearing everybody get back together tomorrow, starting at 8 a.m. Eastern time. So we hope that you enjoyed Day 1, and we are looking forward to Day 2. Thank you.